My acknowledgements to Sunaram. He sent me his slides. Though I have not used, I have used a couple of pictures, but the slides helped me clear my thinking process. So this was not there in his slides. This is necessary post lunch to bring your concentration back. Maybe all the blood is pulling to the stomach. We want some back to the brain. Any idea what she has to do with BCR or TCR rearrangements? Or we'll leave it for later as usual. So what we have seen so far, what is acquired immunity, basics of receptor signaling, antibodies, and the lymphocyte development has been shown very beautifully by Pankti. But the question is, the immune system is stopping thousands, or is it millions? Actually, it is stopping even billions of different types of microbes or any foreign particles. So let's do the calculation. We'll give an example of mathematics. Because you are already sleepy, it will help you fall asleep. Normal person's expected antibody repertoire is 10 to the power 9, 1 billion. Or in computer terms, we call it 1 GB. And if you calculate the number of base pairs in the human DNA, it comes to approximately 1.5. So just think, to make 10 to the power 9 antibodies, one antibody will not be with one base pair. For each antibody, you will need at least 200 to 2000 different base pairs. So around 1 TB of data will be required. But our DNA has the capacity of carrying only 1.5 GB of data. That too, 98% is junk. It is non-coding DNA. So practically we are having 2 MB memory space, and there we are supposed to load 1 TB data. So like the entire matrix database you download and put it on a floppy drive. So this perplexed all the scientists last, since as Pankti had showed in 1965, they showed that the B cell, T cell lymphocytes were different. The B lymphocytes were producing antibodies. That time there was a big confusion. How can the genetic information, which is so less, people at that time estimated there are 30,000 to 40,000 genes in the human genome. But when the Human Genome Project was done, the number was even less. It is now estimated there are 20,000 to 24,000 genes. How can 20 to 24,000 encode for so many different proteins? Then Dreyer and Bennett, they suggested that what they found out from analyzing the structure of different antibodies, they suggested that there would be a constant region of the antibody, which, here, in between. That was their structure. Their structure was not fully defined. So that is what they thought would be the structure of an antibody, and they thought this was the common region, common for all antibodies, and this variable region would be different and coming from a different part of the genome. Again, there was a lot of controversy. People were thinking, how can two different parts of gene come together? This is not mRNA. mRNA splicing is different. You can take one mRNA, you can splice it and make different polypeptides. You can make combination of different polypeptides. But here what they are saying is that two different genes from different places are combining together and forming one protein. So there was a lot of controversy regarding this concept. But ultimately, this gentleman, Susumu Tonegawa, he designed the elegant experiment to show that this is possible. Coming back to what Sanket had shown, the structure of an antibody, simplified 2-dimension, it is like this. We have a heavy chain, we have a light chain. This is the hinge, and the majority of the light chain and approximately half of majority of the heavy chain and approximately half of the light chain, this part is a constant region. So there is not so much variation. But the part, the antigen binding part or the FAB portion, that has high variability. So is it possible that they are coming from different parts of the genome? What experiment was done by Tonegawa, he took embryonic liver, rat liver, because this would show you the germline cells, unchanged cells, unchanged DNA in the fetus or the embryo. And he took radio-labeled RNA against the variable region. He tried very hard to make RNA complementary to the constant region, but most of the polymerases will write from 3' to 5', but he could not make it from the opposite strand. So what he did, he made one for the variable region and he made one for the entire antibody. So you can subtract and find out what is the difference. Today's these experiments can be done in few hours, but he took a agarose plate that was 2 feet long. It took 3 to 4 liters of agarose, and he ran the gel for 3 to 4 days. Then whatever strips were there, he would cut, then melt, and then put RNA, then try to extract what was there. So this was very cumbersome experiment. Nowadays with technology such as SGS page, you can do it maybe within 6 hours. Then what he saw, first he put the radio-labeled RNA against the variable region, and he could see that the band was seen here at one place. And this was, obviously the DNA was it was cut by restriction endonucleases. Then when he ran both the probes, he saw that there were 2 parts. So one was possibly attaching to the variable part, one part was attaching to the constant part, and they were at different places. They were at different places means there is some site where the endonuclease has cut in between. That is why we are getting 2 parts. But ultimately the protein is one. So maybe some recombination has occurred. Then he took a plasma cell producing tumor, a myeloma, from the rat. And with this he ran the same experiment. And when he put the RNA labeled RNA against the variable portion, it was someplace else. So possibly a bigger fragment has become smaller. If it is below means it has traveled more. And when he tried with the bigger one, he again got a single band. So this may be attaching either or both to this, but there was a single band now when he used the larger portion directed against the variable plus constant portion. So what does it tell us? In the germline DNA, there was something in between, there was a site where the 2 parts were separated. But in the B cell, these 2 parts were possibly much together and the endonucleus could not cut in between. There was no restriction site for the endonucleus. This is a complicated experiment. I had to read it 3-4 times to understand it. I tried to simplify it, but in summary, it is that 2 parts of DNA which were previously separate have come together now. For this experiment, it was done in 1970s. He got the Nobel Prize in 1987. And it started off a revolution because first time it was shown that in a somatic cell, this is not a germ cell, in a somatic cell, a very large mutation is occurring. Then people studied the structure of antibody further on, and when they tried to see what are the amino acids, they found that there are extra amino acids here, which were not encoded in this variable region described by Tonegawa, and they called it the joint region. And then again, in the heavy chain, they found another extra region was there, which they called the diversity region. And if we go back to the structure of the antibody, this is the green one is the antigen on this side, and that is the antigen on the other side. And you can see there is a perfect fit between these two. This is the light chain, this is the heavy chain, and you can see what we can do is we can just separate it. This is separation by, I think, 4 angstrom. We can see it is a complementary fit. And these are encoded by complementary determining regions, what Sanket had mentioned. And as you have seen, Pankti had described so nicely the evolution where first a pro-B cell is there, then the pro-B cell rearranges the heavy chain. And once the heavy chain rearranges, it forms a complex with a pseudo-light chain. And there is a pre-BCR. This has the actual heavy chain, but light chain has not yet appeared. So there is a pseudo-light chain, which is called pre-B or V lambda 5. And with these segments, it is expressed, and as she had said, it becomes a double D2. And then there is recombination of the light chain, and ultimately it develops into a mature BCR. What happens? Further development occurs only after there is exposure to the antigen, which will be covered by a subsequent speaker. So when we look at the structure, this is the heavy chain, these are the light chains. The heavy chain has 1, 2, 3, there will be so many variable regions, but only one of these will be chosen. Similarly, there will be various diversity regions, there will be various joint regions. Similarly, in the kappa chain, there are variable regions and joint regions. It doesn't have diversity. The light chains do not have a diversity regions. And then there is the constant portion which encodes for the mu chain. Mu chain means it will end up as a IgM. If it is a delta chain, IgD. If it is a gamma chain, IgG. If it is a epsilon chain, IgE, and so on. So that brings us to the initial question I had asked. That was ABL, BCR. Why did it translocate with 22? Here also we can see some famous lymphomas, Burkitt lymphoma. Here again 8 is there. Then 2 is there, 22 is there. Follicular lymphoma, common is again 14. Mantle cell, common is 14. Lymphocytoplasmic lymphoma, it is 14. What is there in 14? It is the immunoglobulin heavy chain. Because these cells are rearranging, if there is a mistake in the rearrangement, this something else comes and attaches to that chromosome 14, and that leads to all these kinds of lymphoma. Similarly, there can be a break in chromosome 2 or chromosome 22, which are basically the sites for light chain recombination. We saw in Philadelphia chromosome it was 22. So near the, that is BCR, breakpoint cluster region, that also has the gene for the lambda chain. So this may be a reason why when the machinery goes wrong, it leads on to all these kinds of lymphoma. And obviously, we can expect these are all B-cell lymphomas. In T-cell lymphomas, we'll get in the gene where there is TCR gene is located, alpha and beta gene. So, in this mess, how does the cell determine which part is to join with what? You have so many bundles of wire and you have to join them in sequence. So there is something called a recombinant signal sequence. There is a nanomer. Nanomer means 9 specific amino acids are there, and there is a heptamer, 7 specific amino acids. And in between there can be either 12 base pairs or there can be 23 base pairs. These are non-specific, but these two signaling peptides are very specific because they will help to for this to bind. And what is the importance of these base pairs in between? If there are 12 base pairs, it represents one turn of the DNA. DNA, as we know, it is double helical structure, while 23 base pair represents a double turn. So wherever there is a rule that the heptamer will attach with the nanomer, heptamer will attach with heptamer, nanomer with nanomer, and on one side there should be a 12 base pair, On the other side, there should be a 23 base pair. I'll show you the structure why this is so. And the genes are in such a way, you can see this direction, 7 to 9. And this is 9 to 7. So the diversity region has opposite facing. So these two can combine with each other. But all the genes in heavy chain, variable region will be oriented in the same direction. All the diversity genes will be in the opposite direction. So that this variable will not go and attach with another variable part of the gene. But it will go to one part of the diversity segment, and again this diversity segment on the opposite side, it will have the opposite orientation, which will help to attach it to the joint part. Even for the light chain it is same, but obviously there is no diversity region here. And it can occur in two ways. It can the DNA can fold upon itself, and this 7:9, 7:9 will point in the same direction, and this part in between will be cut out and thrown out. Otherwise, it can have a complex which is called an insertional. Then the joint will be there, but the extra part of DNA, which is shown in red, will also stay. 90% is mostly deletional, and you get something which comes out. This is slightly bigger thing. Deletion, you can see the ring will come out, and the 5' and 3' ends will join. And in the inversion, the V and J region will come together, and whatever is in between will be pushed to the left side. So what is the importance of this signal joint? This ring structure is called a signal joint. Any idea? It is used for screening for SCID. So what is SCID? Severe combined immunodeficiency. If T cell is not there, if B cell is not there, it is called severe combined immunity. Severe means cell-based immunity is also not there, and acquired immunity is also not there. And how do you know that T cell development is not there? Means there will be no signal joint formation, and obviously B cell also. So there is a specific RT-PCR targeted against this part, which has been excised. It is excised and thrown out. So the babies at birth, you can do a heel prick, take a blot of the blood on a dried paper, and do a RT-PCR and see. Normally the number of copies should be more than 800, but if it is less than 25, it is highly suggestive of SCID. There are a couple of SCIDs which do not have this, but 95% of SCID will be detected by this method. So US, I think almost 27 states out of 50 states, they are doing the newborn screening for SCID. India also, the option is available. Now coming back to our mathematics, what we have seen in the human gene, heavy chain has 41 variable regions, 23 diverse regions, 6 joint regions. So approximately 5,600 combination is possible. Similarly, 200 for kappa chain and 160 for delta chain. And even if you combine them, we are getting approximately 2 into 10 to the power 6. Around 2 million combinations are possible taking heavy chain, light chain combinations. But what we have talked about initially, we need to find a combination which will explain 1 billion. So it should be at least 500 times more. So where does this extra diversity come from? Again, going back to the complementary determining regions, what we have talked about, antigen, antibody, light chain, heavy chain, there will be the complementary determining region, CDR 1, 2, and 3. Of this, the CDR3 has the highest amount of diversity. And how does this come from? Let us again go back to the monomer, the RSS signal. We can see one strand of DNA is coming here, winding around. This part, the heptamer is attaching, and behind the nanomer is attaching. Then this is a very long chain. They have shown very short, but this will be very, very long. This is a very long loop. Then again it is coming here, On the other side, the nonamer is attaching, and on the back side, the heptamer will attach, and this is the continuation. So on one side, there will be single loop. On the other side, there will be double loop of DNA. And these are held together by a group of enzymes, including RAG1, recombinase, and RAG2, and certain other enzymes. And what they will do, they will excise here. This will become the signal joint, and they will join here. And this is one specific V region, this is one specific J region. And then again, downstream, again, same thing will happen. So what are the other molecules? One is Artemis. Artemis is the Greek goddess of hunting and fertility. Anyone knows her Roman counterpart? Because Roman gods and Greek gods are similar, they have counterparts. Who is the Roman counterpart of the Greek goddess Artemis? Diana. Diana is the Roman counterpart. And Diana, obviously in fiction, not obviously it is mythology. From mythology to comics, Diana has become Wonder Woman. So Artemis, what does it do? When this joint takes place, joining has to take place here. For that, there has to be a cut. So Artemis induces the cut in the folded DNA. The cut can occur over here. So this strand is longer, this strand is shorter. It can be a perfect cut in the middle, or it can cut somewhere here. So one upper strand is longer, lower strand is shorter. Then what will happen? Either these extra nucleotides will be cut out by a exonucleus, because if there are overhanging edges, it cannot be joined again. So there is one endonucleus which can trim this. Or more commonly, there can be introduction of palindromic. Palindromic means mirror image. Mirror image nucleotides can be attached to that. That is by a group of enzymes, of which most important is the DNA ligase 4. And since these are mirror image nucleotides of what is there already on one chain, they are called palindromic nucleotides. Sometimes what happens, especially in the heavy chain locus or in the T cell receptor, we are talking here about the B cell receptor or the antibody. In heavy chain locus of B cell and TCR, there is one extra enzyme available which is called terminal deoxytransferase. So this can do more mischief, and it can add any nucleotide in between. It is active only in the T cells and B cells, and it has a special ability. Whenever it sees a single nick, the DNA strand is doubled, one end is free, then it will go and add any nucleotides of its own. This is clinically useful. We use it in something called the tunnel assay. That is this enzyme, terminal deoxynucleotide transferase, UDP, nick and labeling. Wherever there is a breakage in the DNA, suppose the cell is undergoing apoptosis, there will be breakage. So this enzyme will go and attach some nucleotide, and that nucleotide can be tagged with a fluorochrome. And once we put our light, the fluorochrome will light up. So this is a molecule called DAPI. DAPI will go and attach to all DNA, and this fluorochrome will attach only where there is a break, because of this enzyme will introduce the nucleotide with the fluorochrome. And we get a picture like this. This is a positive control. These are all the nuclei, and almost all the nuclei are having nuclear breaks. This is the negative control. Only nucleus are stained, but there is no staining of the fluorochrome, or there is no light from the fluorochrome. And here in the middle, we can see that apoptosis is occurring in these specific cells. Out of the whole cells, these specific cells are showing apoptosis. So it can give you a quantitative value of how much apoptosis is going on. Coming back to here, what we have seen, small revision, the DNA chain is folded, Artemis will cleave here or here or here. Suppose it is cleaving here, there is an overhanging edge. To complement the overhanging edge, the mirror image nucleotides will be added. These are called P nucleotides. P nucleotides means palindromic nucleotides. And other strand also same thing is happening, P nucleotides. And when TDT is there, it will randomly put in some nucleotides here. And these are all non-coding nucleotides. Since these are non-coding, they are called N nucleotides. So one is N nucleotide, one is P nucleotide, and sometimes what happens, this is just cut off. Overhanging edge is just cut off, that is nucleus. And for this group of enzymes are important. DNA ligase 4, XRCC4, Q70 and Q80. Q70 and Q80 are also called XRCC5 and XRCC6 and Cernu knows. Why am I mentioning this? Because all of these enzymes have some clinical importance, like REG deficiency. If the REG enzyme is not there, there cannot be recombination of the receptor genes, and there will be no T cell, no B cell, no development. Then Artemis also, if it is absent, it can cause KID. This DNA ligase will cause KID plus other mental retardation, microcephaly, other features are also there. But the rheumatologist may be more interested in Q70 and 80. Why so? Where do we get Q? In overlap myositis. So overlap myositis, there are antibodies against Q. So anti-Q antibodies, they will, this again, as you can see, this is a cytosolic DNA receptor. So whenever cytosolic DNA receptor, tomorrow again Pankti will talk about it in details, when there is a false signal, you can just think of lupus as a viral infection. Fever, skin rash, joint pain, you get it whenever you have a viral infection. Arthralgia. So you can think of a prolonged, complicated viral infection. Why are you having that infection? You are not actually having an infection, but the body thinks that infection is going on. Because this is a cytosolic DNA sensor or some other sensor is there which is giving a false signal to the body that there is exposed DNA, double-stranded RNA. So the body thinks this is a virus and we have to eliminate it, and the inflammatory process can manifest as myositis, lupus, Sjogren, whatever. Then coming back here, so we can see maximum of this occurs when there is the combination. One combination is between V and J, then J and D, then D and, V, J, J, D and D, C. So wherever there is combination at each of this site, you can get so many new nucleotides. And whenever you get new nucleotides, you end up having either a new protein or there will be a frame shift mutation, stop codon, then there may be no protein formation also. But all this junctional flexibility, addition, and the exonuclear section will add to the diversity. And so when we talk about diversity generation, our initial calculation we had got 2 into 10 to the power 6. Then there is something called combinatorial diversity, combinatorial diversity. So each B cell will have 2 heavy chains and they will have 4 light chains, 2 kappa, 2 lambda. So any 2 of these can attach with any one of the 4. So another 8 combinations we are getting here, and rest of the variability is introduced by P nucleotides, palindromic ones, N nucleotides, non-coding ones, and exonucleates trimming. So this is what happens in the immature but developed B2 B cell. Once there is exposure to the antigen, there is something called somatic hypermutation. Again, that is post-antigen exposure. That is a upcoming talk. Only one question remains. There are 2 heavy chains and 4 light chains. What if the cell starts to rearrange and produce proteins from all? If two different B cell receptors are there, that will cause lots of confusion. So the cells have a intrinsic mechanism by which they will allow only one heavy chain. As soon as one heavy chain matures, the other VGD recombination is inhibited. I'll just show you the picture. So this is what is happening. Initially, in the pro-B cell stage, these are two probes. One, they have put a probe against the variable region, once against the constant region. So there is a gap between these two. This is one locus and this is the other locus because two alleles will be there. Then in the pro-B cell stage, what happens? The DNA unwinds and forms this kind of loops. There will be Reg, Artemis, and DNase ligase here. And these two will compete. One of them will recombine first. If it recombines first, then it will inhibit the recombination of others. So when it reaches the pre-B cell stage, these two have stayed together because the recombination has occurred. But these two, as soon as this recombination occurred, it will inhibit the other one, and these two have separated back again. So once there is successful recombination of one chain, the second chain recombination will be inhibited. If the first one fails, then obviously the second one will get rearranged. And if the second one also fails, because after recombination, there is no 100% guarantee it will form a functional protein. If there is a frame shape mutation, or if there is a stop codon, then they will not form a heavy chain. So the cell will die out. So initially to make heavy chain, the cell has 2 chances, and to make light chains, the cell has again 4 chances. So this is just depiction of that. First try successful, very good, it will go into pre-BCR, then it will change over to light chain. If first try is unsuccessful, it will try the other heavy chain. This is unsuccessful, it will go and die. If second one is successful, then again it will try to, initially always the kappa chain is tried, one kappa chain failed, then other kappa chain will be tried. If that also fails, cell will die. If it is successful, it will go out. If it is not successful, now the lambda chain can be tried. If lambda chain is successful, it will go out. Otherwise, the other lambda chain will be tried. So the cell has 8 different steps where 8 different combinations can be tried out. And ultimately, if nothing works, then the cell will go to death. So the efficacy of this is around 55%. Here also 45% of the B cells will die. And of those who survive, again, Pankti has showed 95 of them are dying. So this is such a energy consuming process, but the body wants everything to be perfect so that we do not have autoimmunity. That is why we can see almost 98 to 99% of the cells which have, of the B cells are going and ultimately dying. Is there a second chance? In the first movie, Terminator, Schwarzenegger was the villain. In the second one, he has become a hero. Nowadays, it is usually opposite. Hero becomes villain in subsequent movies. But there is something called light chain receptor editing. So sometimes what happens, what Pankti was showing, they will have a positive selection. That means cell is surviving. Then it will show the self-antigen. And if the self-antigen it is being shown, and again it is matching there is a what you call negative selection. It is matching a self-antigen. Then the cell has 1 second chance before dying. Suppose the kappa chain was there, it can go down, it can change the kappa chain and present a different kappa chain. So this is a concept of light chain receptor editing. Doesn't occur with heavy chain. Initially, heavy chain will be there with the pseudo light chain. It will be expressed. Then once the light chain is expressed, heavy chain light chain first BCR will be expressed. In case it reacts with self-antigen, the cell has only one chance to go back and redo try another combination of the light chain. So this is one mechanism which it can save the B cells. Then how does it produce IgD, IgA, IgE? The switch from IgD to IgM is simple. That occurs at the level of mRNA. I'll show that. And the switch to IgA and IgE is basically a class switch recombination. And this occurs after antigen presentation. So thankfully, I don't have to take care of it. A more classy ma'am will come and tell you about class switch recombination. This is basically the mRNA slicing. If it slices at one particular position, the mRNA will go and produce IgM. If it slices at a different position, it will go and develop a IgD. Immature cells will have more of membrane IgM. Mature cells will have more of membrane IgD. And after exposure to antigen, then again it will shift back to secretory IgM. Time is short, so I will not go into details, but the TCR story is practically the same. Instead of having a heavy chain and light chain, here we have a B chain. B chain is just like the heavy chain for antibody, and we have a alpha chain, which is like the light chain of the antibodies. So same story overall. I'll take a shortcut, and only major two differences are there. In the V delta gene, there can be two diversity regions. Instead of VJD, it can be VJDD. So there is slightly more diversity. Usually the gamma delta don't have too much diversity, but the delta gene, which is synonymous with the light chain, they can have two diversity regions to increase the diversity. And they do not undergo somatic hypermutation. What is somatic hypermutation? Again, after antigen contact. And what I have told about allelic exclusion. If one B cell receptor is there, it will not allow another B cell receptor. This is not 100% true in case of a TCR. 30% of T cells may have two TCRs, two different types of TCRs. And obviously, there is no secreted product. In the B cell, the receptor is secreted in the form of an antibody. Here, it is not secreted. That is why understanding the TCR took 20 more years. BCR is very easy. It is secreted. You can take a myeloma, and the myeloma will produce lots of antibodies. You can understand the structure of the antibody. But the TCR is stuck to the cell. You cannot separate it from the cell. And if you break the cell to isolate it, you will get thousands and millions of proteins. That is why the history, that is very interesting. But obviously, time is not there. And post-last session, I don't think much more will be digested. So what we have seen, DNA can change in mature cells. This is somatic mutation. This occurs normally in healthy individuals, but as Sir had mentioned, it can also occur in pathological states in specific cells and lead to certain phenotypes of disease. We are just beginning to understand that. That is going to take maybe another 5 years, 10 years to know about this somatic mutation in diseases. Most common MCQ related to this is how to generate diversity. One is the combination of the segments, then 2 heavy chain versus 4 light chain, then the mirror image palindromic nucleotide, the extra by DTT, that is non-coding nucleotide, and extra nucleotides can be excised by the exonuclease streaming. So the next speaker will be coming to talk about MHC. Antibody diversity is at an individual level, but MHC diversity will be at a community level. So I will have only maybe 18 to 20 different types of MHC, but approximately 1 billion different types of antibodies. We have seen how diversity occurs at the antibody level and what is the implication, how this helps even for a TCR in the presentation that will be covered by Chengappa next.